Hello and welcome to an intro to protein synthesis. To understand protein synthesis, we must first understand what DNA is. Deoxyribonucleic acid, or DNA, is the material that contains all the genetic code for most organisms. It is found in both the nuclei and the mitochondria of each cell and referred to as nuclear and mitochondrial DNA, respectively. It is essentially a recipe book with the instructions for making all the proteins in our bodies. The recipes are stored in blocks called nucleotides, which are made up of a ribose molecule, a phosphate molecule, and one of 4 nitrogen bases: adenine, thymine, guanine, or cytosine. The nitrogen bases pair up with each other, A with T and C with G, to form the familiar double helix structure. The helix is arranged to have a beginning called 5' and an end called 3'. The two strands run anti-parallel to each other. The sense strand runs 5 to 3, and the antisense strand runs 3 to 5, and acts as a template during the transcription process so the mRNA molecule can accurately copy the correct sequence.
What is RNA? There's often a misconception that DNA is responsible for all the work when it comes to the inheritance of genetic traits. However, it wouldn't be possible for genetic information to be transferred without the work of RNA. RNA, or ribonucleic acid, is a nucleotide that is essential for roles of coding, decoding, regulation, and expression of genes. The main differences between RNA and DNA is that RNA has ribose sugar, whereas DNA has deoxyribose sugar. RNA uses uracil to pair with cytosine, whereas DNA uses thymine. RNA is also single-stranded, where DNA is double-stranded. There are actually 3 types of RNA, and we will break them down to gain a better understanding of how they come into protein synthesis. 1. Messenger RNA, known as mRNA, is responsible for taking messages from chromosomes to ribosomes using the information provided by DNA. tRNA, known as transfer RNA, is responsible for bringing amino acids together to build proteins. Each tRNA has an anticodon on one of its ends, which pairs with a complementary codon in the mRNA. Ribosomes are made up of rRNA, or ribosomal RNA, which form ribosomal subunits to help build proteins.
So now we understand the difference between DNA and RNA. But how do they work together to make proteins? This happens through a 2-step process called transcription and translation, which we'll talk about in more detail soon. DNA holds all of the genetic information for the coding of these proteins, but in order for this information to be shared, it has to go through a process of transcription. This is where it essentially creates a copy of its unique code and an mRNA molecule is formed. mRNA, or messenger RNA, has a very important role in protein synthesis. It has the ability to leave the nucleus to deliver all of the required information into the cytoplasm, where it attaches itself to a ribosome, the protein-making factory, and coordinates with other cellular organelles and molecules to read and translate this information to create proteins. tRNA, or transfer RNA, is another key molecule involved in this process. Attached to each tRNA molecule is an amino acid, which are the building blocks for proteins. Each amino acid has a unique code called an anticodon that binds to a complementary codon on the mRNA strand. Once enough of these amino acids have linked together in a certain sequence, a protein is formed and released into the cytoplasm. Now let's learn a little bit more about this process in depth.
Transcription begins in the nucleus, where the enzyme RNA polymerase binds to DNA and unwinds the strands in order to read the bases of the template DNA strand in order to create a complementary sequence of RNA bases. These bases are then bonded together to form mRNA. After mRNA has been processed through various methods, it exits the nucleus and travels to the ribosomes in the cytoplasm. Ribosomes contain important structures called rRNA, which contain 3 binding sites for tRNA: the A site, the P site, and the E site. The A site accepts the tRNA carrying an amino, the P site is where the peptide bonds form between the amino acids, and the E site is known as the exit site, where the tRNA leaves the ribosome. The ribosome reads the codons on the mRNA and tRNA, brings a corresponding anticodon and amino acid. Elongation factors move the mRNA down the ribosome structure one codon at a time, and the tRNA attached to the ribosome moves from one site to another, known as translocation. Translation is terminated when the A site encounters a stop codon and the polypeptide chain is released from the ribosome.
Central dogma is the outline of the progress of information in cells. Central dogma explains that DNA codes for RNA and RNA codes for proteins. According to central dogma, the sequence of bases in an organism's DNA is what determines its genotype. The phenotype is product of the proteins the organism produces. Although some alleles share the same gene, their DNA sequence differs. Therefore, the amino acid sequence in different alleles of the same gene is likely to be different also. Some exceptions do occur to the central dogma. Many genes do coding for other RNA molecules such as tRNA and rRNA that do not function the same as mRNAs and therefore cannot be translated into proteins. There are some instances where the information flows backwards from RNA back to DNA. This is seen in some viral genes which use reverse transcriptase and are composed of RNA. An example of this is HIV.